Developmental networks show a collection of gene products that can interact with each other. Each gene product in the network either inhibits or promotes the transcription of another gene product in the network. Today we are going to be looking at a developmental network in the spiky dragon. Let's think of this developmental network as a map to a special treasure that we wish to obtain, spiked cells. Each obstacle on our map represents a specific gene product in the network, which sequentially affects whether or not we are going to be able to obtain our treasure or transcribe our spiked gene at the end. On our wild-type map, we see two gene products, or obstacles, that are inhibitory. These obstacles prevent us from starting the next obstacle. For example, obstacle P inhibits us from moving on to obstacle I, and obstacle I represses obstacle K. We see three obstacles that are promoters. These obstacles bring us closer to our goal and promote us on to the next obstacle. Let's look at our map to determine if our wild-type developmental network would lead us to a spiky cell product. Starting out, we see that gene product S promotes us to obstacle or gene product P. However, obstacle P is inhibitory, repressing us from moving on to obstacle I. I usually has the ability to repress K, but in this case, because I itself is repressed, it cannot function to repress K, so K is freed. With K unrepressed, we can make it to the end of our pathway and get a spiked dragon. What if we change the obstacles present in the developmental network? If the developmental network lacked a functional P gene product, then I is no longer inhibited. We can make it to that obstacle. If I is expressed, then that means it will be able to inhibit K. With K inhibited, we can no longer be promoted to obstacle E, and our dragon will not have spikes. What if our developmental network lacked obstacle I? Well, we discussed before that obstacle I inhibits K. If I is removed, K is no longer repressed, and we are promoted to obstacle E, and then promoted again to our treasure, giving our dragon spikes. What if our developmental network lacked both I and E? Well, with I out of the picture, we said that K can be expressed. That's a good thing. However, we still need E to be expressed to promote us to our treasure. Without E, we cannot reach our last step to transcribe the spikes, and our dragon will be spikeless. If we instead removed S and K, we would see that S can't promote P. If P is not promoted and transcribed, then I will not be inhibited. K is gone, however, meaning that we will never be able to promote to obstacle E and never be promoted to the spiky treasure. So, now that we understand how developmental networks work, let's think about a different application regarding our spiky dragon. Let's assume that all of our spiky dragon cells have the same genome. However, even though they all have the same genome, some of the cells appear spiky and some do not. Why is this? Well, one type of regulation that may be at play is DNA methylation. In the course of life, aging processes, environmental influences, and lifestyle factors such as smoking or diet induce biochemical alterations to the DNA. Frequently, these lead to DNA methylation, a process in which methyl groups are added to particular DNA segments without changing the DNA sequence. DNA methylation functions to regulate transcription. It tends to cause genes to be turned off. These tend to occur at the start site of transcription. Methylation of the genome in these regions attracts deacetylases, making it more difficult for other transcription factors to come along later and activate transcription. Therefore, if a portion of one cell's DNA is tagged with the methyl group, the DNA that encodes for spiky cells may not be transcribed and therefore not expressed as a spiky phenotype. Even though the cell has the same genome as the cell next to it, if the cell next to it is not methylated, the unmethylated cell will be more likely to be transcribed as usual and result in the spiky phenotype.